RNA virus. An RNA virus is a virus that has RNA, ribonucleic acid, as its genetic material. This nucleic acid is usually single-stranded RNA, sRNA, but may be double-stranded RNA, dsRNA. Notable human diseases caused by RNA viruses include Ebola virus disease, SARS, rabies, common cold, influenza, hepatitis C, hepatitis E, West Nile fever, polio, measles, and coronavirus disease 2019. The International Committee on Taxonomy of Viruses, ICTV, classifies RNA viruses as those that belong to Group 3, Group 4 or Group B of the Baltimore classification system of classifying viruses and does not consider viruses with DNA intermediates in their life cycle as RNA viruses. Viruses with RNA as their genetic material, which also include DNA intermediates in their replication cycle, are called retroviruses, and comprise Group V of the Baltimore classification. Notable human retroviruses include HIV-1 and HIV-2, the cause of the disease AIDS. Another term for RNA viruses that explicitly excludes retroviruses is ribovirus. Characteristics: single-stranded RNA viruses and RNA sense. RNA viruses can be further classified according to the sense or polarity of their RNA into negative sense and positive sense, or ambisense RNA viruses. Positive sense viral RNA is similar to mRNA and thus can be immediately translated by the host cell. Negative sense viral RNA is complementary to mRNA and thus must be converted to positive sense RNA by an RNA-dependent RNA polymerase before translation. As such, purified RNA of a positive sense virus can directly cause infection, though it may be less infectious than the whole virus particle. Purified RNA of a negative sense virus is not infectious by itself, as it needs to be transcribed into positive sense RNA. Each virion can be transcribed to several positive sense RNAs. Ambisense RNA viruses resemble negative sense RNA viruses, except they also translate genes from the negative strand. Double-stranded RNA viruses. The double-stranded, dsRNA viruses represent a diverse group of viruses that vary widely in host range: humans, animals, plants, fungi, and bacteria, genome segment number 1 to 12, and virion organization, triangulation number, capsid layers, spikes, turrets, etc. Members of this group include the rotaviruses, renowned globally as the most common cause of gastroenteritis in young children, and picoviruses, renowned worldwide as the most commonly occurring virus in fecal samples of both humans and animals with or without signs of diarrhea. Bluetongue virus is an economically important pathogen of cattle and sheep. In recent years, remarkable progress has been made in determining, at atomic and subnanometeric levels, the structures of a number of key viral proteins and of the virion capsids of several Rna viruses, highlighting the significant parallels in the structure and replicative processes of many of these viruses. Mutation rates. RNA viruses generally have very high mutation rates compared to DNA viruses, because viral RNA polymerases lack the proofreading ability of DNA polymerases. This is one reason why it is difficult to make effective vaccines to prevent diseases caused by RNA viruses. Retroviruses also have a high mutation rate even though their DNA intermediate integrates into the host genome, and is thus subject to host DNA proofreading once integrated, because errors during reverse transcription are embedded into both strands of DNA before integration. Some genes of RNA virus are important to the viral replication cycles and mutations are not tolerated. For example, the region of the hepatitis C virus genome that encodes the core protein is highly conserved, because it contains an RNA structure involved in an internal ribosome entry site. Replication. Animal RNA viruses are classified by the ICTV. There are 3 distinct groups of RNA viruses depending on their genome and mode of replication. Retroviruses, Group I, have a single-stranded RNA genome but, in general, are not considered RNA viruses because they use DNA intermediates to replicate. Reverse transcriptase, a viral enzyme that comes from the virus itself after it is uncoded, converts the viral RNA into a complementary strand of DNA, which is copied to produce a double-stranded molecule of viral DNA. After this DNA is integrated into the host genome using the viral enzyme integrase, expression of the encoded genes may lead to the formation of new virions. Classification. Classification of the RNA viruses has proven to be a difficult problem. This is in part due to the high mutation rates these genomes undergo. Classification is based principally on the type of genome, double-stranded, negative or positive single strand, and gene number and organization. Currently there are 5 orders and 47 families of RNA viruses recognized. There are also many unassigned species and genera. Related to but distinct from the RNA viruses are the viroids and the RNA satellite viruses. These are not currently classified as RNA viruses and are described on their own pages. A study of several thousand RNA viruses has shown the presence of at least 5 main taxa: a levivirus and relatives group, a picornavirus supergroup, an alphavirus supergroup plus a flavivirus supergroup, the Rna Viruses, and the Strand viruses. The lentivirus group appears to be basal to all the remaining RNA viruses. The next major division lies between the picorna supergroup and the remaining viruses. The Rna viruses appear to have evolved from A plus VRNA ancestor and the VRNA viruses from within the Rna viruses. The closest relation to the V-stranded RNA viruses are the Rna positive strand RNA viruses. This is the single largest group of RNA viruses with 30 families. Attempts have been made to group these families in higher orders. These proposals were based on an analysis of the RNA polymerases and are still under consideration. To date, the suggestions proposed have not been broadly accepted because of doubts over the suitability of a single gene to determine the taxonomy of the clade. The proposed classification of positive strand RNA viruses is based on the RNA-dependent RNA polymerase. 3 groups have been recognized. A division of the alpha-like, synbus-like, supergroup on the basis of a novel domain located near the N-termini of the proteins involved in viral replication has been proposed. The 2 groups proposed are the altovirus group, alphaviruses, foroviruses, hepatitis E virus, hordeviruses, tobemaviruses, tobraviruses, tricornaviruses and probably rubiviruses, and the typovirus group, apple chlorotic leaf spot virus, carlaviruses, patex viruses and timoviruses. The alpha-like supergroup can be further divided into 3 clades: the ruby-like, tobema-like, and timo-like viruses. Additional work has identified 5 groups of positive stranded RNA viruses containing 4, 3, 3, 3, and 1 orders, respectively. These 14 orders contain 31 virus families, including 17 families of plant viruses, and 48 genera, including 30 genera of plant viruses. This analysis suggests that alphaviruses and flaviviruses can be separated into 2 families, the Togaviridae and Flaviridae, respectively, but suggests that other taxonomic assignments, such as the pestiviruses, hepatitis C virus, rubiviruses, hepatitis E virus, and arteriviruses, may be incorrect. The coronaviruses and toroviruses appear to be distinct families in distinct orders and not distinct genera of the same family as currently classified. The luteoviruses appear to be 2 families rather than 1, and apple chlorotic leaf spot virus appears not to be a closterovirus but a new genus of the Patexviridae. The evolution of the picornaviruses based on an analysis of their RNA polymerases and helicases appears to date to the divergence of the eukaryotes. Their putative ancestors include the bacterial group 2 retroelements, the family of Triproteases and DNA bacteriophages. Proteoviruses are related to and may have evolved from a totivirus ancestor. Hypoviruses and Barnaviruses appear to share an ancestry with the potivirus and sobomavirus lineages respectively. Double-stranded RNA viruses. This analysis also suggests that the Rna viruses are not closely related to each other but instead belong to 4 additional classes: Rnaviridae, Cystoviridae, Proteoviridae, and Rheaviridae, and 1 additional order, Totiviridae, of one of the classes of positive Rna viruses in the same subphylum as the positive strand RNA viruses. One study has suggested that there are 2 large clades. One includes the Calyciviridae, Flaviviridae, and Picornaviridae families, and a second that includes the alpha Tetraviridae, Rnaviridae, and Cystoviridae, Notaviridae, and Permutotretraviridae families. Negative strand RNA viruses. These viruses have multiple types of genome ranging from a single RNA molecule up to 8 segments. Despite their diversity, it appears that they may have originated in arthropods and to have diversified from there. Satellite viruses. A number of satellite viruses, viruses that require the assistance of another virus to complete their life cycle, are also known. Their taxonomy has yet to be settled. The following 4 genera have been proposed for positive-sense single-stranded RNA satellite viruses that infect plants: Rna Virus, Rna Virus, and Virus. A family, Sarthriviridae, which includes the genus Rna has been proposed for the positive-sense single-stranded RNA satellite viruses that infect arthropods. Group 3, Rna Viruses. There are 12 families and a number of unassigned genera and species recognized in this group. Group 4, positive-sense Rna Viruses. There are 3 orders and 34 families recognized in this group. In addition, there are a number of unclassified species and genera. Satellite viruses. An unclassified astrovirus/hep virus-like virus has also been described. Group B, negative-sense Rna Viruses. With the exception of the hepatitis D virus, this group of viruses have been placed into a single phylum, Rna Viruses. This phylum has been divided into 2 subphyla, Haplovirucotina and Polyplovirucotina. Within the subphylum Haplovirucotina 4 classes are currently recognized: Chungquayu Viruses, Milne Viruses, Monji Viruses, and Yunchang Viruses. In the subphylum Polyplovirucotina 2 classes are recognized: Elia Viruses and Instoviruses. 6 classes, 7 orders and 24 families are currently recognized in this group. A number of unassigned species and genera are yet to be classified. Gallery Notes. The majority of fungal viruses are double-stranded RNA viruses. A small number of positive-strand RNA viruses have been described. One report has suggested the possibility of a negative-stranded virus.